going to talk about some of our platform's aims, just so that any of the new folks are aware and that are clear in like the constraints that we have placed on this particular platform. Typically, I usually kind of have this lecture from the standpoint of microbes. And so just to be clear, microbes are bacteria, viruses, fungi, small, tiny organisms that you have to see with a microscope. So that's usually like what we call a microbe. So usually I talk about those a lot, but I don't talk a lot about cells. So today's kind of lesson, I'm going to use cells to communicate my point. And so just to start, so when we ask, like, why are we interested in cells and microbes? What I want you to take from this is we're interested in cells and microbes because those are topics that are relevant to us and to our everyday, right? All living organisms are made of cells. And so this means anything from an animal all the way to a plant, all of them are made of cells. And then even all of these microbes, except for viruses, all of these microbes are themselves made of cells, except it's usually just one cell, right? A virus is usually not considered a living organism, even though it's a microbe, because a virus requires a host in order to live, right? So that's usually the distinction, and that's usually what I mean when I use these terms. So let's kind of give a quick backstory on cells and who discovered them and how they learned about them. So cells were first discovered in 1665 by a British scientist called Robert Hooke. So what he did is he was looking at cork cells and looking at how they appeared under the microscope. So then he saw that the way that what he saw when he used a microscope was he saw distinct kind of structures that looked almost like small cells. I believe the story says like the cells in a monastery, like the organized structure where someone can go now to pray. That's what he thought of, and I believe that's how he came up with this idea of cells. And one thing that I learned when I was preparing for this is I didn't know that cork is made out of. So cork is like the kifuniko of the wine bottle typically. I didn't know that that is made out of the bark of a plant. So honestly, I learned today. I thought that was like some manufactured thing, but it's actually from a plant. Anyway, so this is what he was looking at, and that's where he saw these cells, and that's what he was really interested in. Let's see comments. So that's what he was interested in. Hello, Hannah. Thank you, Asante, asante. Anne Wera says, Karibu sana, Team TAUKIRWO. Thank you. Okay, so sorry. So I was saying that he looked at cork under the microscope, and that's when he saw the distinct cells. And then in the 1950s or so, the electron microscope allowed scientists to see smaller things inside the cells. And so this means that the structures that are located inside the cell became easier to visualize and see. And then in the later in the 1950s, the structure of DNA was discovered. And it was discovered by Francis Crick, James Watson, Maurice Wilkins, and Rosalind Franklin. All of them together did this work that led to an understanding of what is DNA specifically. Maybe one day we should do a lesson about what is DNA specifically. So that might be in December. Anyway, so these folks, they kind of pushed our knowledge forward and our understanding forward into understanding the specific structure and the specific organization of DNA. So up here you see the people themselves. Down here you see their paper that they published in 1953 that was talking about the molecular structure of DNA. And then here on the bottom right, what you see is the famous picture that was taken by Rosalind Franklin. So she used this technique called X-ray crystallography. And so what happens is you prepare whatever molecule you want to understand, and you prepare it into a crystal. So think about like a sugar crystal, like a solid structure. You prepare it like that. Then you shoot basically an X-ray beam at it, and then you look at the way that that beam has diffracted, and then you can back calculate the organization of the molecules inside that crystal based on the diffraction pattern. So what you're looking at here is a diffraction pattern, and then basically this team was able to now back calculate the molecular organization of DNA, and that's what really supported the kind of discovery of the way that DNA looks like. So that happened now in the 1950s. But I should also stress that in 1869, there was a biologist from Switzerland who first isolated nucleic acids from living things. So you might be asking yourself, okay, now how have we gotten to DNA now? But the reason why we to think about DNA in this topic is because DNA is the instructions basically that the cells use to live and to make their decisions and to make their choices and in how to like stay alive, basically. I want you to think about biological cells almost like a small city. And inside that city, there are specific instructions, kind of like the law. And those instructions are the ones that the city uses to know how to navigate itself. And those instructions are typically DNA. And that's very important to think about when we are thinking about like the cell and how the cell functions. Because if you remember, some time back, we talked about cancer and we talked about how sometimes in the disease of cancer, cells grow without an order and without organization. And so some of that disorder comes from a mistake or an error or something that has changed in the way that these instructions from the DNA are followed, right? And so that's kind of how all of this ties together. And that's what we are interested in, and that's what we talk about here. And that's the perspective that we use in all of our lessons. And then I think later in time, I want to stress that, so now we are basically kind of walking through history and walking through discoveries that helped us understand what cells are, how they look like. I see there's some comments. Let me pause. Okay, I'm here to learn about cells and also microbes. I'm really, really glad that you're here to learn about that. What is the difference between cell and microbes, please? That's a really good question. So, okay, when we say cell, that just means a structure that is like the foundational structure of a living thing. So, a cell just generally is an organized unit that is a living unit, basically, right? So us as humans, we are made of different tissues. Those tissues are made out of different cells, and all of those cells working together are what allow us to continue living and to stay alive. A microbe is basically another organism that is usually they're unicellular. And now those organisms infect cells, but like you can say a bacteria. So let me back up. A bacteria is one cell, right? And that one cell can live outside of the body. It can also live inside the body and give us infection. It can now infect now the cells of the intestine, for example, and then it messes up those cells and then that gives us disease. I hope that has given you some clarity. So a cell is a general term for a living unit, like the foundational living unit. And then a microbe is a specific type of cell that usually lives outside of like a big body, if that makes sense. And so, so now when you think about a fungus, that is another living unit. A bacteria is a living unit. An amoeba is a living unit. And then those units can now infect the living units in us that make up our tissues, organs, and that make us the way that we are. I hope that has provided some clarity. Let me know if that did. Do single cells belong to plants? So plants, so in the same way that we are made of many cells, plants are made of many cells. Following from Kayole 1. Thank you. So happy to have you here with us. What are the benefits of microbes? Maybe the next topic you can teach about how beneficial microorganisms in animals and plants. Yes, that's a great topic. We will write that down. What are the benefits of microbes? So sometimes microbes, I guess I can use them in the context of like our gut. So we have some microbes that live in our intestines, and sometimes those microbes can help us digest food better. They can provide different things that help those cells live better and function better. There's one type of bacteria that lives in our gut that actually helps us make vitamin K, which is important for the way that we live. So those are some of the benefits of microbes. And that kind of theme is present both in animals and even in plants. I think in animals, I can think of this one example. There's a type of Vibrio called Vibrio fischeri that lives in a squid that lives in the ocean. And what's really interesting is that these Vibrio, they fluoresce at a certain time. And that's really important to help the squid because sometimes that's important for like the environment that this squid kind of exists in. So there are different ways that microbes work with the organisms that they live in, and that can be important for their lifestyle and their livelihood. So I hope that answered the question. But yes, that's an excellent topic. I think we'll definitely focus on that next time. So yeah, I think I answered those questions. Okay, so now where are we? So we've basically been talking about different discoveries that have been made over time to help us understand what we understand about cells. So the last discovery I wanted to kind of talk to you about. So instead of just telling you cells are important, I decided to show you what we've been learning about them and what we've learned and how the discoveries over time have helped us kind of build upon itself. Okay, so now what you're looking at right now is a jellyfish. So in the 1960s, sorry, someone asks, do all cells have DNA? Yes, all cells have DNA because DNA is basically the chemical instructions that dictate what happens inside that cell. So, okay, sorry. So now where are we now? We're now in the 1960s. So around the 1960s, people started thinking about technologies and techniques that can help see and understand what's happening inside the cell, and most importantly, follow proteins inside the cell. So if you remember, the slide before, okay, so I talked about how in the 1950s, the electron microscope allowed scientists to see smaller things, right? And then in now the 1950s, the structure of DNA was determined. And so people now are starting to take some steps into understanding the biological cell, right? And then I stressed that in 1869, there was a biologist from Switzerland who first isolated nucleic acids from living things. So we are just kind of like moving along key components that help us understand the cell and key discoveries that biologists have used to understand the cell better. Okay, so now we are in the 1950s. And so now in the 1960s, like I said, they started to have some questions about, okay, what is made by this DNA? So this DNA makes RNA, the RNA makes proteins. What are these proteins doing? Where are they inside the cell? So those were some of the questions that people were asking. And so then one of the technologies that was developed or that was really important to kind of move now this problem along was to be able to look at some of these proteins using artificial tools. And those tools have little fluorescent signals that you can now use under a microscope, right? And those fluorescent signals and those fluorescent chemical tools, they actually came from nature, from jellyfish. So this jellyfish that you've been looking at, I think I've had this up for a long time, but this jellyfish that you've been looking at, what happened is that there was a scientist who we'll talk about in a second, who noticed that this was a fluorescent jellyfish. And so he wondered, can I use this tool to understand questions about biology? So he harvested the tools and he was able to use, I think actually this jellyfish was luminescent. And so he harvested a protein from it that had this luminescence. Then he applied that to understand different questions in an experimental context. Later, from other types of jellyfish, other fluorescent, other tools that could allow you to see, specifically called fluorescents, those were harvested and developed. And then so those were like tools that now people could use to look inside living cells. And so this is a very important detail because you can imagine it's one thing to look at a cell under the microscope after you've killed it, after you've fixed it. But it's another thing to look at living systems and kind of follow different molecules in those living systems. And so actually that led to these people. So the first person who kind of started this was this person, Osamu Shimomura, and then there are several people after him who now developed and evolved and moved forward these fluorescent tools that are now used in modern biology and that are so important. And so they won the Nobel Prize in 2008 for their work in developing these tools that helped us understand how the cell works. And specifically, this means by allowing scientists to take pictures using microscopes and then seeing with their eyes where these different proteins exist and where they are. And that's a very important detail. And so let me show you a picture of our cells. These are heart cells that were taken by by a scientist who studies the heart. His name is Dr. Serrano. And he took these pictures. And what I want you to focus on is the fact that there are all these different colors and that they are organized and located at different points in this cell structure, right? So what happens is that these small fluorescent signals are attached to specific chemical targets that allow you to kind of bind to different parts of the cell. So then together you can kind of look at it all at once. And that's what you're looking at here. You're looking at an organization of cells from our heart. And so what I want to kind of stress here is that over the last few decades, there's been a lot of discoveries and progress to help us understand more about the cell and more about living cells, how they function and how they work. And this is kind of the context in which we talk here on this channel. Here on this channel, we talk about discoveries and different topics. We talk about them from the side of diseases. So we usually talk about a disease that people understand, and then now we talk about the cells and the microbes that are involved in that disease. But today I wanted to kind of share with you a little bit about some key discoveries that have helped move the field along. So I see some questions. So let me pause and ask. Another question, do cells reproduce? Yes. So cells reproduce by dividing themselves. So if you remember in our cancer lesson, we talked about how cancer is formed when the instructions for dividing go crazy, right? So the instructions, so typically what happens is that there's a very specific order and structure that organizes that like process. And during a disease like cancer, those instructions and those orders, they are no longer listened to. So the cell just keeps dividing. And then that's why you end up with this mass, you know? So yes, cells reproduce. If you watch the cancer video, you will see the one that's on the channel. I actually talk about some of the process of the reproduction process and the division. The reproduction is specifically like a division. And so I talk about those in molecular detail. I know some people told me that was too much detail, but Anne, if you're interested in more details, please, I recommend that you hop onto there and listen to that. And please let me know if you have any questions. So we have that on the channel now. So let's see. Joan Mawere says, watching, I'm really glad you're here. Thank you for being here. Learning, I'm happy that you're learning. That is what we are here for. I am happy to see that. Let us see. There's another question. I saw kuna cells in a complication that can cause disease. How do they? Okay, so cells do sometimes repair some of the problems that they have. But let me think. So, okay, tell me the level of detail you want. Do you want a lot of detail or do you want an overview and then I give you links to read more? Because I noticed that there are sometimes I make videos that have too much detail and then people get a bit frustrated. So, maybe we'll make that topic and then we'll do it live so that you guys can ask questions and give feedback. Maybe that's what I'll do. Yes, so we'll put that on our list for December. John, we'll put that on our list for December. We'll talk about the way that cells repair themselves and the way the systems and the techniques that scientists have understood and have developed to kind of understand those questions. We'll put that on December's list. So do cells repair themselves? They have ways to help themselves. Usually, like they'll kill themselves. Sometimes, like if there's certain things that are going wrong, they do have repair mechanisms. We can discuss them. Yes. This is a very long answer to say yes. We can make a video focused on how cells repair themselves. I heard that there are cells that are never replaced. Is that true? Yeah, there's some cells that once they die, they don't come back. Then there's other cells that they're called stem cells. They can be regenerated and they can kind of come back. So yeah, there are different types of cells have different properties. And yes, this is one such property. There are some when they get sick, that's it. Okay. So now we are talking about the tools that have been developed to look inside cells. And those tools are fluorescent. And so that's why we can see this picture. And I think that was the extent of the lesson. So I think the purpose of this small journey was to tell you that we like to talk about cells because we are made of cells and because there's a whole field of people that spend their time understanding how these cells work and how these cells function. Send also an article we can learn, but if possible, you do a live discussion. Yeah, of course, I can do that. Okay, so we've talked about how people understand cells, and I've given you a very brief history of some of the key things that people have understood about cells in the last, in the historical understanding that we have about cells. And we know that the reason why we care about cells is because we are made of cells, and a lot of the understanding, a lot of the current understanding of disease is based on these premises, right? Because there are certain, whenever you're taking a medicine, usually it's trying to kind of counteract or amplify a process that's happening at the cellular level. So that's why this topic is important, and that's why it's kind of relevant to disease and our understanding of disease. And let's see, I think honestly that's it. So then the last thing was to do with this idea of like, what is the platform's aims? Because we've gotten some new people. So the aim of this platform is it's an education platform. Basically, what I'm trying to do is provide tools and resources to understand basic concepts related to disease and related to health, but from the standpoint of what's happening at the cellular level. And we do that by focusing on the different cells in the body and also the microbes that infect the cells. So that's usually our perspective. And I think that's the reason why we have that perspective is because my background is in the biomedical sciences. And when you have a PhD in this field, typically you're focusing on the tiny details. So that's why you get these tiny details. Like that's why the focus is that way. However, if I was a different type of health professional, meaning someone maybe with an MD or a DO or a BSN or an NP, if that was my perspective, then maybe the organization of this platform would be a bit different in that maybe now one can focus on the human aspect of things and the health aspect of things. However, that is not my expertise, that is not my strength, and I don't have the training for that. And so that's why this is a perspective that you get. I've gotten some questions that ask me about that. And so I wanted to clarify that. So the reason why the platform is structured the way that it is because of my background and my training. But I still think that it's important for people to have this understanding, because the reality is that usually, like when you're in the hospital, things are very rushed, and the doctor may or may not have time to explain certain concepts. And you may or may not have the background to understand the concept. And because we know doctors, I think, are usually really stressed out and rushed, and health professionals usually have a lot of stress on themselves. Oftentimes, like very key concepts sometimes can get missed. And so that's really what we are trying to do. We are trying to fill in like this general scientific education gap so that you have a little bit more information when you're dealing with certain health challenges. And I think that makes the whole process a bit easier, because now you're starting with a little bit more awareness and a little bit more education before you need to go to the hospital, before you have a health emergency. And so that's what I'm committed to providing, is like this basic education, basic science education. And that's why I'm here. That's what I'm here for. Okay, so Tabi Shiro asks, germs and microbes, are they the same? So yeah, that's let me think about this. So typically when we say germs, that's usually associated with disease, right? So I think germs is a word that specifically discusses microbes that cause disease. However, not all microbes cause disease. So I think I would kind of distinguish in that way. I would say that germs almost always will cause some type of problem, whereas a microbe may or may generally, not all microbes will cause disease, you know? So, yeah. Sorry, I had to sneeze, and then I lost the sneeze after I muted myself. Okay, so let's go to the next one. Germs are bacteria, viruses, fungi, and protozoa. Some microbes are also the same. Does it mean zinamaanisha kitu moja? So germs are those microbes, but usually related to disease. But then microbes is the big family. Just basically a microbe is something that you need a microscope to see. And it's usually external of, you know, a host or a body, if that makes sense. So yes, the microbes are the same, but not all microbes cause disease. Oh, I'm happy. Thank you. Thank you for your question. Okay, so I think honestly that was same thing. I guess some germs are good for one to keep us. Yeah, so yeah, I think because you know we exist with these microbes, we exist with these germs, right? So it's good to kind of be exposed to them because they help to train your immune system so that your immune system is aware and it's not always reacting and it doesn't get sick. So yeah, they are part of our being alive. Microbes are part of being alive, right? So and that's why we talk about them, and that's why they're important, and that's why they're relevant. Because I think sometimes people, they come onto this channel and then they imagine that, the topic is distant and it's not related to them, but this topic is very relevant. Microbes are important and essential to our everyday life, and they show up in our everyday life. Okay, so I think that was all I had for today, honestly. It was just another reiteration of what are cells, what are microbes, because a question I got recently is someone wanted to know why we are called Tunyamotwe Moyo and why we don't talk about like the body and health and disease. Like why is the title of the channel not something related to the body or health? But I wanted to stress today that the reason why that is because our emphasis and our focus is on the little Tonyamoto Totwemoya, the little living organisms. And then what I would like you guys to do is to now use that information and have it in hand when now dealing with disease and when thinking about now the larger context, meaning how now this exists in us. So that means that hopefully after being on this channel for a while, next time you have a virus and then someone tells you, oh, just go take antibiotic, you can tell them, no, but a virus is not the same as a bacteria, so an antibiotic can't help me. So that's the level of, I think, knowledge that I would like you guys to have, and that's why I want you guys to have it, you see? So that's basically that. And then someone says, please, in your list, do for us the importance of cells and microbes. With that, we will learn more. So actually, Tabi Shiro, if you're here, I would like for you to ask specifically what you would like me to say about cells and microbes. Because I think so far we have lots of different perspectives. So in the past, I've talked about why we care about them. Today, I talked about why we care about them, but I gave you kind of like a historical context of how we learn about them. So I would like to know, like, what specific question do you have about microbes and cells that you would like for me to explain and to discuss? And once I see that, then I can willingly make that video. And also do for us about infection if possible. Okay, that's a good question. But what about infection are you interested in? Are you interested in like how our immune system responds to infection? Are you interested in the different mechanisms of infection? Because so what's interesting is the field of cell biology, molecular biology, virology, microbiology, all of those fields, quite seriously, what people are doing in those fields is understanding infection. So that means for every bacteria that you can imagine, it has its own way of infecting. And now that's the job that scientists like me do, is we understanding, okay, how is this bacteria, whatever bacteria you're working with, how does it infect? And that's literally the job. And sometimes when you see someone with a PhD in some of these fields, that's what they did. That was their contribution. And then the same with a virus. We understand, okay, viruses infect us, but there's people whose work is how. What proteins are made by the virus? How does it infect? The same with a fungus, the same with a protozoa, all of these systems. So this is to say, Tabi Shiro, when you say about infection, what specifically would you like to learn? Would you like to learn about the way that a specific microbe infects? And if so, what type of microbe would that be? Because imagine each one has its own system. So like the bacteria I study, it's called Shigella. So the way that it infects us is it has this type 3 system, or it's called a type 3 system. It looks like a little syringe. And that syringe is what it uses to attach to the cell, and then it injects its poisons and its things into the cell. So that's the mechanism that it uses. It's called a type 3 secretion system. And that's a key part of its infection. A different type of bacteria has a different system, a different mechanism. A virus has a different mechanism. So this is to say, infection is a very, very big question. And so I'd like to know what specifically about infection you might be interested in. Okay. So infection of common cold, of a UTI. Yeah, we can talk about that specifically. So I'll put that on the list. So we will talk about what causes the common cold. It's typically a virus called the rhinovirus. And we can talk about how it infects and how it is transported, transferred, and how it kind of goes around in our body. And then the same as a UTI, like what is a UTI? How do you get it? How do those usually UTI is a bacterial infection, if I remember right. So we can talk about what are the common ones there. Okay. And also stomach flu. Okay, we'll add that to our list. And STIs also, please, I know a lot will back up on this. Of course, yes. So we have on our list for December HIV, and the HIV virus is the one that causes the disease AIDS. So we have that on our list. I think someone had asked about the bacteria that causes gonorrhea, so that can also go on the list. So maybe we'll just have a list of different bacteria that cause commonly spread STIs, and then we can focus on that for a lesson. Yes, that is easy enough to do. We will put that on our list. Georgia James, I support this also STIs. Yes, yeah, we can focus on STIs because again, like in the thing that I think if you remember when we were talking about meningitis, remember we said meningitis is spread by like mucus from our nose. So I think in that lesson we talked about how like all sorts of fluids and secretions from our bodies can be vectors or can be ways in which these microbes, i.e., these bacteria, these viruses, how they can be spread from person to person. And so this means all sorts of secretions from all different body parts, including the ones that are involved in sexual communication, sexual interactions. And so that is how those get spread. And we can definitely focus on those. And what I'm trying to think about is whether you guys want, like, okay, maybe you can tell me while you're here. Do you want to focus on like one disease one at a time, or do you want to focus on like the general topic? Or do you have specific STIs you really want to know about? Maybe let's do that. Do you have, what is the, like maybe the top 3 that you want to learn about? How infection works. Okay, so we can make sure to include that. So for every topic we discuss, we will focus on like the infection mechanisms. So like on the day that we talk about HIV, we can say, okay, this is the part of this virus that allows it to infect. So when we talked about COVID a long time ago, we talked about the fact that the SARS-CoV-2 virion, it binds the specific receptor in the lung cells, and this is the way it enters. So yeah, we can make sure to include that detail in our STI lecture. That is easy enough to do. Okay, you want general topics. Okay. General topic, all diseases. Okay, so maybe what we can do is we can organize that lecture into STIs general. Generally, we can say, okay, STIs as the name communicates is a disease that is transmitted via sexual contact. And within that family of diseases, there can be these bacteria, these viruses, and then we can maybe talk about how each of them infect and maybe what they are, what they look like. Maybe I think I've not put a picture up in a while, so maybe we can look at some of those pictures, and then we can talk about, yeah, like the specific details of how they infect. That's easy enough. We can plan for December to do that. Okay, types and cause and treatment. Okay, so the treatment part is tricky because I'm not a medical doctor, so I try very hard to stay away from giving any specific medicines and any specific treatment, but maybe we can talk about generally. So for example, for some of the bacterial ones, usually there's antibiotics, although there are some that are antibiotic resistant, and so that's why one has to be really, really careful and stay safe, because for some of them, there's antibiotic resistance. And if you want to learn more about antibiotic resistance, there's a video on my channel about what is antibiotic resistance. So maybe we can highlight that then. But yeah, that's easy enough. We will make you a lesson and we will post it, and we will plan to do that in December for sure. Treatment tutaona daktari. Yes. Yeah. So I won't give specific treatment, but I can talk about general. Like we can talk about the families of treatments that people typically use. Maybe that's a safer thing to do. We can do that. Okay. So I think that is it for today. Let us see. I think oh man, I've not been looking on OG Media's So is anyone on OG Media's Facebook page, and are there questions there that I have forgotten? I have not forgotten questions. Okay, let's see. I think not. I think I'm able to see all of the questions here. Thank you. Esther Maina, you are so welcome. Thank you for being here today. Okay, so I think I have answered all the questions. If I have somehow missed your question, what I will do after this live stream is over is I will go back and look at the comments again to make sure that I have answered all of the questions. I do think I have them all. Yeah. Okay. All right, you guys. Thank you so much for being here. Thank you for being patient with me as I figure out how to stream this properly. Hopefully next time, the third time will be the charm and I will do this properly the first time. But thank you for being here. Thank you for coming here and asking your questions. And thank you for consuming the lessons that we make and for answering the questions and for for asking questions, and thank you for giving me the opportunity to answer the questions that you guys ask. All right. Bye.